Hi there, welcome to lesson 7.10. This lesson will cover herbivorous or plant parasitic nematodes, and will specifically look at Heterodera avenae, which is the cereal cyst nematode. And the image on the left represents Heterodera avenae females protruding from an infected root. Okay, let's jump into lesson 7.10. So let's look at phytophagous nematodes. So phytophagous means plant eating. These are all the nematodes that consume plants, usually at the root level, and they do so through the various stylets and spears that they possess as part of their mouth parts to actually pierce plant cells and plant tissue. So these devices normally divide into the telenchoid stomatostyle or the dorilomoid odontostyle. And all that means is basically that these two families of nematodes possess certain features of the stylet or the spear. And you can see some of the examples in here, but these all belong to the same genus Heterodera, but the species do have variations in them, particularly in that stylet or spear region. Most are migratory ecto or endoparasites, so they either live on the outside of the plant root or inside of the plant root, and only some are sedentary where they do not move. They might be polyphagous, so that means consuming various plant species, as opposed to being host-specific, where they will only parasitize one plant species or a family of plants. And really, species feeding habits depend on the ecological settings. So while there are generalizations being made, each nematode and the plant it is associated with, the extent to which parasitism will occur depends on the ecological settings in that particular soil, even more specifically within that particular relationship. So that particular plant with that particular nematode. And of course, this is rarely looked at, and yet that is exactly how these organisms need to be studied in order to really understand why that relationship occurs in the first place and the extent to which the parasitism takes place. So let's look at the various groups of plant parasitic nematodes. These divide into the sedentary parasites. An example of that is the Cereal Cyst nematode, which we'll describe in a minute, and that's the Heterodera genus. Another genus we'll describe is the Meloidogine, and that's the root knot nematode. There are also migratory endoparasites, so they live inside the plant, but they do migrate, and Pretilinchus is another one we'll talk about as well. So there are also semi-endoparasites. So that means that part of their life cycle can be lived out outside of the plant. And there are ectoparasites. So these don't actually require the plant tissue to live inside of. They feed externally on the plant. And there are also epidermal cell and root hair feeders. So they specialize in just feeding on the epidermal cells of the roots and the root hairs themselves, as well as piercing moss feeders. So these specialize in feeding on mosses. And this beautiful diagram here on the right just shows the relative sizes of the major genera of the plant parasitic nematodes. And the ones we'll focus on in particular are the Heterodera as well as Meloidogine. And these actually are cyst nematodes. So they form cysts and they do not look like all the other nematodes, as you can see here. And what is noteworthy is the differences in size in the different nematodes. So we can see here Pretilinchus, which we'll also talk about, is very tiny in comparison to some of the larger plant parasitic nematodes. So these are the three groups that we'll look at because they are the major agricultural parasites of crops, and they are the most economically significant out of all the different genera and species of plant parasitic or phytophagous nematodes. So plant parasitic nematodes are in fact an agricultural phenomenon. They do not really cause problems in polycultural ecosystems where there are many different plant species available. But in monoculture, they are certainly an issue, and particularly here you can see their effects on cereals. So this is in wheat plants in particular. And because cereals supply 20% of calories to the human population worldwide, and there's an ongoing decrease in production versus an increasing demand for cereals, it is a major concern to the agricultural industry that grows cereal crops. Wheat production needs to actually increase by 60% to fulfill the estimated demand that's going to be there for wheat by 2050. And plant parasitic nematodes decrease world crop production in general by 10%. It is one of the primary causes of wheat yield losses worldwide, and parasitic nematodes of wheat have been estimated to cause $5.8 billion worth of damage in 1987 and $173 billion per year worldwide by 2019. So the major pest of wheat in particular, or other cereals, is the Cereal Cyst nematode. So we're going to use the acronym of CCN for that, but the genus is Heterodera, and there are several different species in that genus. There's also root lesion nematode, which we'll call RLN for short, and that is the Pradolincus genus, which is distributed globally. So we can see here that cereal cyst nematodes cause this strange appearance of roots in cereals. So this knotty appearance, so the roots are stunted, they do not develop as well, and here we have a root that's filled with female nematodes which are in their cyst form. So the Heterodera genus displays some plant family specificity and sometimes plant species specificity. So there are different Heterodera species that affect different plants. So for example, there's the sugar beet cyst nematode, that is a specific species, the cereal cyst nematode, but that affects many different cereals. And this is the one we'll be talking about. That's Heterodera avenae. There's rice cyst nematode. So that's a Heterodera that is specific to rice varieties. There's the pigeon pea cyst nematode, soybean cyst nematode, maize or corn, clover, and the brassica family of plants also has its own Heterodera cruciferae species that prefers brassicas as its hosts. In the image on the right, we have the various features of the soybean cyst nematode, Heterodera glycinus. And this is the cyst, which is always the female that actually contains the eggs. And the genitalia of the various species of Heterodera are very specific, and this is how the different species are identified based on these features of the female cyst. This is an egg that has the juvenile nematode developing inside. This is an infective juvenile called J2, and this is the sugar beet cyst nematode, showing the difference between the features of the soybean cyst nematode as opposed to the sugar bean cyst nematode. So it appears that the cereal cyst nematodes, or cyst nematodes in general, seem to be the animals that have the most studied vaginas in all of the animal kingdom. I don't know if that's actually true, but that's what it seems like when we look at the extent that goes into investigating the various species of nematodes based on the features of their vulvas. And these are the vulvas of the avenae group. And you can see the various species and the differences in the various structural features of the nematode vulvas in the different species. So it is not uncommon that many different invertebrate species specifically have their genitalia studied in order to identify them and distinguish the species within the genera. And this diagram just shows the various morphological features of the nematode's vulva in order to identify the species. So there are these particular distinguishing features in the avenae group that allow investigators to know which species of Heterodera is present in the field, consuming the crop. It is super interesting to see how structurally complex this part of the nematode is, and its usefulness not in just producing eggs but also in species identification. So in addition to the Heterodera genus being divided into various species, some of the species themselves represent groups of species. So for example, Heterodera avenae is in itself a complex of 12 different species of the Heterodera avenae group. And this table shows the different species of the Heterodera avenae complex. And as you can see here, some of these species affect cereals and grasses, and some grasses only, but not cereal crops. To further understand this table and how the Heterodera species are divided and their economic impact, etc., you can have a look at this interesting plant disease publication called Cereal Cyst nematodes in the Western USA. So all of the nematodes that belong to the Heterodera avenae complex or group of nematodes are sedentary endoparasites of over 200 plant species. It is the females that are sedentary, even though the males also have a sedentary part of their life cycle. They are motile and can move, but the females have a life cycle within only the one plant, and they form these hard brown cysts with eggs. And these cysts persist in soil and give rise to the eggs, as well as the juveniles that hatch. And this mostly occurs at the root zone between 5 and 25 centimeters within the soil. But these cysts can be present anywhere from the surface of the soil to up to 60 centimeters in depth. As such, they are spread easily by machinery, animals, and water. And the hatching of the infective juveniles is dependent on temperature, soil moisture, and in some species also depends on root exudates. The infective juveniles enter the roots and feed on cortical cells of the plant, and there might be between 1 and several generations per season depending on the species. And this is a beautiful image of Heterodera avenae juvenile with the gorgeous piercing mouth part, the stylet that allows it to penetrate through the plant root. So we will now specifically describe the cereal cyst nematode, as these are obligate parasites of various crops, and there are those 12 species that affect cereals and grass roots. The most important economically are the Heterodera avenae, latipons, and Filipjevi. Heterodera avenae can cause cereal yield losses across Western Asia, North Africa, Europe, Australia, and the USA. And wheat-specific yield losses vary between 90% in Saudi Arabia to 10% in China. And here we have the Heterodera avenae worldwide distribution map. So basically it affects pretty much all continents, and the most prominent species in terms of their economic impact are listed here, and these are the various countries within which they exist, but they are all part of this group of Heterodera avenae complex. And this is just a general life cycle. It's nice and simple, even though it is not of a cereal cyst nematode, but most of the Heterodera species have a similar life cycle. And the way these nematodes proliferate is through this cyst. So the female becomes a tough brown cyst, protecting the eggs for long periods of time, making the nematode persist in the fields. The cyst contains lots of eggs once it comes out of the plant, and each egg has an individual nematode that then hatches under specific conditions. And this is the J2, or the juvenile infective stage of the nematode, that is able then to migrate or move until it finds a host plant, and then it will penetrate and get into the plant tissue where it is able to actually induce the plant to produce a group of cells that become this feeding site, and then they become stationary in there and feed on those particular cells, and a lot of the plant's resources will go into those feeding sites. So this is why there is a yield loss in the crops upon which these nematodes feed. And while this stationary feeding occurs, the nematodes actually go through various stages of development by molting, and eventually the male becomes mobile or motile and moves out of the plant to mate with females, while the females actually stay stationary and become a cyst and protrude out of the root. And these females, while developing, they will get fertilized. Some of them don't need to be fertilized, but in some species they do. They will produce eggs, and those eggs will develop inside the female. Some of them form this gelatinous matrix where the eggs are deposited outside as well. Some of the species will not do this, but in fact the female will just die and become the only egg-containing reservoir. And once again, the cycle begins once the cyst opens up and releases the eggs, and the cycle continues on as already described. So this is the general Heterodera life cycle. And now we'll get into more details in relation to the cereal cyst nematode. So now let's specifically look at the Heterodera avenae life cycle. And we'll begin with the infectious second stage juvenile hatching from the egg, and then being able to find a plant and use the powerful mouth part to pierce the root cell wall and get into the plant root and move its way into the root cortex. Once inside the cortex, the juveniles induce the plant to form the syncytia. So these are the feeding sites that are formed from multiple cells that become one multinucleate cell, and the juveniles become sedentary there and are able to just feed on this syncytium here. These are the juveniles in pink, and they become sedentary and keep feeding until they become the fourth stage juveniles, and this is the last stage before the males leave the plant and are able to move on to other plants, whereas the females remain stationary and become white cysts protruding from the root. And once this occurs, they are ready to be fertilized by males, and the egg maturation process begins. And inside one female, there can be between 100 to 600 eggs, but this depends on the species of the nematode, the host plant, and the soil type and the environment. Heterodera avenae specifically does not produce the external egg sac like other species do, and they only have the one generation per year, as well as not being stimulated by exudates from the plant. So basically, Heterodera avenae will hatch in the absence of hosts, which may reduce its survival and persistence since there will be no plants for the next generation of nematodes to feed on. But once the eggs mature, the female dies and turns into a brown cyst that then falls off into the soil and is filled with matured eggs. And these eggs will eventually hatch, releasing the infective juvenile, and the cycle continues. So this is the specific Heterodera avenae life cycle in a nutshell. So let's take a closer look at the life stages of the cereal cyst nematodes. So in image A, we have eggs that are ready to hatch, and you can see the second stage juveniles ready to go and leave the egg. Here, the J2 has hatched inside the cyst and is actually exiting the cyst. Here we have a J2 that has been extracted from the soil. Here we see it probing at a root, trying to get in and piercing the root with its large hypodermic needle to get into the root, and once in the root, it migrates through the root cortex to a site where it can develop a syncytium, so a feeding site, and it does so by inducing the plant to use its cells and produce this one big cell with multiple nuclei to feed the nematode, and this happens via secretions produced in the pharyngeal glands of the nematode. Then it molts and becomes a J4, so the final stage of its development. And once matured, the female, which is featured here, becomes this white cyst that protrudes from the root tissue. And if fertilized, she's filled with eggs. These are some young encysted females harvested from the roots. These are rolled cysts that are collected from the soil. And finally, we have a cyst that has been crushed, showing the contents, which are the eggs and the J2s. The Heterodera avenae life cycle requires 3 to 4 months to complete, and a temperature of between 5 and 15 degrees Celsius. And hatching is regulated by temperature and soil moisture conditions. The J2 emergence actually requires dormancy, not in all of the species, but Heterodera avenae does require dormancy, which is called diapause. But this is just dependent on specific species and climate conditions. Hot and dry conditions induce diapause, whereas reduced temperature and increased moisture activate J2s to become infective. And here we've got an image of a cyst that is burst, and all the eggs have spilled out, including some already hatched juveniles. So diapause is a reversible suspended development stage or dormancy, which occurs during unfavorable conditions. And eggs in a dormant cyst may remain viable for several years. And the dormant encysted J2s exhibit very minimal metabolic activity just to keep them alive during that period of dormancy. Not all cyst nematode species exhibit dormancy. And there are 2 different types of dormancy or arrested development dormant state, and that is diapause versus quiescence. So quiescence is reversed with favorable conditions, whereas diapause actually requires more than just favorable conditions to be present. In some species, it is an obligatory part of the life cycle. In some, it is a facultative condition that depends on environmental stimuli, for example, day length. Heterodera avenae requires 8 weeks at low temperature for substantial hatching to occur, and hatching continues at between 5 and 10 degrees Celsius during spring. In the image on the right, we have the beautiful cysts of Heterodera avenae. So cereal cyst nematodes adapt to local conditions and environments in different regions of the world. They synchronize hatching with favorable host plant invasion conditions and nematode reproduction. So for example, cereal growing regions of South Australia, Victoria and Western Australia exhibit similar climates but vary in how the nematode responds to that particular climate. So all have this Mediterranean climate where the summers are dry and hot and winters are cool and wet. In South Australia, there are 2 hatching phases. There's a larval development phase at 10 degrees Celsius and juveniles emerge at 20 degrees. Whereas in Western Australia, the larval development occurs at 10 degrees and juveniles emerge at 15. So that's just to show how the particular conditions, even in particular soil, can change the response of the nematode and its ability to infect the host plant. And here we have a root with female nematodes protruding out of the root. So there have been various attempts to estimate the yield reduction caused by cereal cyst nematodes with regards to the population density or the numbers of nematodes in the soil or number of cysts. However, it is difficult to actually make a connection between original nematode numbers or the nematode load and its relationship to economic threshold at which they become detrimental to crop yield. So these generalizations are difficult to make because they are quite specific to each individual farm. And this is because there is an interaction of climate, crop variety, management practice, nematode distribution and density in the field, as well as chemical, biological, and physical soil properties that affect the intensity of the infection within a specific field. Additionally, drought and inadequate nutrition, soil compaction, and adverse temperatures have a significant impact on how the infection proceeds and the severity of that infection in terms of crop yield. Specific wheat or other cereal variety and its ability to replace damaged roots is also important. So how the plant responds really varies. and depends on the cultivar and once again on very individual field soil-based characteristics. In the image on the right, we have a 4th stage juvenile feeding from a syncytium and some female cysts protruding from this affected root. So there are several management practices that are used in order to control cereal cyst nematodes. So first of all, nematicides are applied as broad-spectrum fumigants, and they are nervous system toxins. So they are very detrimental to all life forms, including human life, and are therefore not advisable to be used and are being phased out. In addition, they are quite expensive, so they are not economically viable. A 70% reduction in nematode infection can be gained by post-harvest rotation with crops such as carrots, fenugreek, and onions. So these are crops that do not support the nematode, so therefore they are non-host crops. So that is a good way of reducing nematode loads and survival. Another method is to leave the field fallow and introduce summer ploughing. So leaving the soil bare without plants is quite destructive to the soil ecosystem, and ploughing is also detrimental to soil microbial life. So therefore these practices actually in the end reduce plant resistance by weakening the soil ecosystem. Soil solarization using polyethylene sheets is another method, but that cooks the soil ecosystem. And some people are using now biocontrol agents such as fungi and bacteria. And this begs the question as to why these bacteria and fungi do not exist in the soil in the first place, indicating that there is a lack of predatory and other mechanisms to naturally control the nematodes. So this indicates once again that there's an issue with the actual soil ecosystem being damaged in some way, making the plants unhealthy and therefore allowing proliferation of the nematodes. Planting winter cereals during autumn is a good method because it allows for deep root development before peak hatching. So this allows for greater root proliferation and therefore more resistance to the nematodes. Planting susceptible trap crops that attract the nematodes before hatching and then killing the plants before egg maturation reduces nematode loads. And ensuring optimal plant nutrition and soil moisture is the best method because both limiting nutrients and water increase crop losses. So really when the plants are happy in an optimal soil ecosystem, nematodes actually are not an issue. And so the very fact that cereal cysts and other nematodes are such a problem is because the soil ecosystem has been damaged. It is overexploited, as are the plants, and therefore the nematodes are basically a symptom of diseased, unhappy plants and soil ecosystems. Here we have an image with various nematode cysts at various maturation stages, and some roots that are infected with young females. So a big area of research goes into creating or finding resistant cultivars, and these plants can actually suppress or prevent reproduction of nematodes, or they are plants where the infective juveniles cannot find a suitable feeding spot on them. In some cultivars, the J4 female development is suppressed. In some, there is delayed syncytium development, so therefore nematode feeding is compromised. In some of these plants, the syncytia degrade, and therefore there is no feeding site left in the plant for the nematodes to feed from. And these plants are developed from resistant wheat germplasm, and the breeding of these plants occurs from wild wheat relatives. And so far, 9 single dominant resistance genes have been identified. However, some of these cultivars, even though they are resistant in one region, can be fully susceptible in another. And that's because these organisms are so sensitive to temperature and moisture and all these other environmental conditions that govern their life cycle. And so this brings me back to this concept that it is always dependent on the actual individual ecosystem within which the plants are growing. Because if the plant is resistant in one soil but fully susceptible in another, it suggests that the problem is not in the plant but actually in the soil ecosystem. Also, the fact that all of these plants come from wild resistant relatives of the plant suggests that these plants in nature are not naturally susceptible to nematode infections. And so therefore there is a problem in the way we treat the plant as well as treating the soil ecosystem within which it grows. Otherwise, these plants should be naturally resistant to the nematodes or allow the feeding of the nematodes to a certain extent, but not to the extent where the plants are completely decimated. And we'll talk more about this when we complete this unit as to how these plants behave in natural ecosystems as opposed to agricultural ecosystems in response to this economic pressure that is put on them in order to generate profit rather than just to grow crops for human consumption. Here we have a third stage juvenile feeding on a syncytium in the root, and these eggs contain first stage juveniles. So we have now come to the end of lesson 7.10, and in our next lesson we'll continue looking at herbivorous or plant parasitic nematodes, but we'll discuss the root knot and the root lesion nematodes. So you'll be hearing from me again in lesson 7.11. Bye for now.